Good morning, everyone. Thank you for joining us. You are currently joining a live feed from the remotely operated vehicle SuBastian aboard the research vessel Falkor, operated by Schmidt Ocean Institute. My name is Deborah Smith. I'm a marine technician on board, and we're just getting settled on the seafloor. We just reached the seafloor about 5 minutes ago. We're currently at 3,685 meters, and we are diving on a vent field in the Gulf of California in the Pescadero Basin. And scientists will come on and do some audio commentary here intermittently when we have some interesting things to talk about. We may also have a few other guest speakers. Please feel free to send your comments and questions on YouTube and Facebook, and we want to welcome you along on ROV SuBastian's 200th dive. So thank you very much for joining us.
Good morning, everyone. We're here on the RV Falcor using the ROV SuBastian. This is our last dive of the series in Pescadero Basin. It's a special dive. It's actually dive number 200 for the SuBastian sub. And right now we're in a hydrothermal vent mat area, so sediment covered area, and we're using some gas tight samplers to collect pristine water samples in the shimmering fluids next to the mats. As you can see in the foreground, when we were zoomed in, there was a bunch of little red organisms running around. Those are actually copepods, and so this is kind of an interesting thing. We were measuring the temperature just right in those fluids where the copepods were concentrated, and it was upwards of over 70 degrees Celsius in the highest part of the flow, and about 40-ish degrees where the copepods were sitting in the depression. So they're definitely enjoying a little warm bath here on the seafloor. The ambient temperature here at 3686 meters water depth is about 2 degrees Celsius, so a little colder than your refrigerator. So there's some pretty spectacular temperature gradients going on locally near the seabed where these focus flow is happening. We're going to take some push cores here to couple with our temperature probe measurements and gas samples, and then we'll move on over to a place we're calling the P vent a little later. It's going to be a shorter dive today. We're going to pull a little bit earlier so we can start our transit back into shore. Good morning, everyone. We are here in Pescadero Basin on our last dive of this cruise. We are at a place we're calling the Z Mound on the flank, collecting some push cores and hydrothermally active sediment. If you've been watching along, you actually saw the previous push core we collected. We inserted it into the hot shimmering water, actually melted, so the core liner got very distorted. These core liners are made of polycarbonate, and their melting point is about 150 degrees centigrade. So it's pretty hot sediment below. Earlier this morning we were doing temperature probe measurements, and the fluids emitted from some of these shimmering waters were close to 90 degrees, just coming out right out at the seabed. So it's pretty steep temperature gradients here. We're going to see how the other core fared, and then maybe move on to another place we're calling the P Mound, which is just a little bit north of us here. So thanks for watching. If you have any questions, feel free to write in and we'll try to answer them the best we can. I'm Victoria Orphan. I'm a co-chief scientist of this cruise, faculty member at Caltech, along with Rob Zerenberg, who's a faculty member at UC Davis. We've had a really wonderful series of dives here, Pescadero Basin. This is dive number 200 for the ROV SuBastian, and we've had a really wonderful time exploring these new hydrothermal vent ecosystems over the past couple of weeks. So thanks for watching. All right, so we had a few questions about what the white organisms were in the mat. Those are actually sulfur oxidizing bacteria, we think. So they're actually about maybe 100 microns in length. So they're among some of the biggest bacteria that we see, and you can tell this by being able to actually visualize them on the seafloor. These organisms typically gain energy by using hydrogen sulfide that's coming from the hydrothermal fluids, and they couple it with oxygen in the overlying seawater. So this is really a perfect environment for these bacteria in this vent site. The other organisms we've been seeing in high concentration here that was a bit of a surprise were these little red little insect like organisms. Those are actually copepods, and so these are organisms that are common zooplankton in the upper midwater ocean. This is a special benthic variety, and we don't know if it's a new species here or not, but they're clearly favoring the warm fluids that are coming out of these hydrothermal vents. So we'll try to get a couple of close-up shots of these copepods before we leave the site if you want to take a look.
For those of you who have checked in online, we are at 3,686 meters depth in Pescadero Basin, which is in the Gulf of California. And we're out here with a team of biologists that are interested in the various organismal communities. So we have some microbiologists on board who are currently sampling the seafloor. And we have several people studying larger animals that live in these very unique habitats. What you're not seeing in this image is all of the sort of explosive underwater volcanoes that are nearby, venting water temperatures at around 290 degrees Celsius. And out on the flanks of those structures is what you see now on your screen, which are these lush microbial communities and some animals. We see these amazing copepods and sometimes some worms that live in these sediments side by side with these sulfide oxidizing bacteria. And you see one of our lead scientists here is taking some push cores in the various colors of bacterial mats. And if you were watching about a half an hour ago, you saw us melt one of those cores, which is sturdy enough to withstand 150 degrees Celsius. So it is very hot where these organisms are living, even on the surface, which was 90 degrees. And you see we're doing a little sideways push core here, trying to get some of this crust that's on the top of this microbial community. And once we're done sampling, we'll be inserting a high temperature probe to try to really get a specific temperature on the depths in the sediments. And we also came back to this site. We've been there once before on this expedition. And we came back because there was quite a bit of hydrocarbons or orange oil that was floating out of the seafloor once we took our cores. And so we're back to take a few more of those samples, and then we process late into the night, sectioning the sediment and trying to measure all different kinds of chemical parameters, including methane and sulfide, pH, and then we'll also assess the microbial community using molecular techniques like sequencing of DNA and then also some microscopy.
Hello everybody, I just want to say hi. My name is Deborah Smith. I'm a marine technician aboard the research vessel Falkor. I work full-time on the vessel along with two other technicians and a number of other crew. We have officers and engineers and chefs and stewardesses. And I just want to say a quick shout out to my niece Abigail who's watching, and just say hi and happy Thanksgiving tomorrow for everybody. Thank you for coming along on our 200th dive with the research vessel Falkor and ROV SuBastian, operated by Schmidt Ocean Institute. I know there have been a lot of questions about the vehicle and what we're doing, and scientists will jump on and do some explanation, as well as our lead technician, Leighton Raleigh. And so just send in your questions on YouTube and Facebook on the Schmidt Ocean Institute pages, and we can answer them live here in the control room. Thank you very much.
Hello and welcome back. We're currently conducting a dive 200 of ROV SuBastian. The vehicle is currently at a depth of 3,685 meters. Ambient water temperature is about 2 degrees Celsius. Some of the vents that we've been looking at here are more like 280 degrees. At the moment the ROV is sat on the seabed and we're taking core samples of the sediment around one of these smaller vent features where the hot water is bubbling through the seabed. The white stuff you see on the seabed is bacterial matter. ROV SuBastian is a remotely operated vehicle operated by Schmidt Ocean Institute. It's connected to the mother vessel, Falco, which is 2.2 miles above it, via a steel armored umbilical. Through this umbilical we send electricity to power the vehicle, but we also send commands via fiber optic and receive the cool videos that you're seeing at the moment. Once on board, the video is then transmitted ashore via a satellite connection. In the control room we have several scientists who are monitoring the operations, as well as a ROV pilot and co-pilot. The co-pilot is currently controlling the manipulator. On board the vessel we have a smaller exact copy of the arm. As the co-pilot moves the replica of the arm, the main arm on the vehicle, known as the slave, mimics those actions. You can imagine controlling an arm 2.2 miles away. As well, in the control room we have a marine technician who is taking care of the data logging. As we go through this, the ROV SuBastian is collecting millions of data points each dive. So far this expedition, I believe we've collected something like 330 million data points just in sensors and values from the vehicle and ship alone. This is the last dive of the expedition. This expedition has been split into two legs. The first leg of the expedition was a mapping portion. Here we were using a number of tools. Firstly, we could map with the vessel, but we also used an autonomous underwater vehicle, which kind of looks like a big yellow torpedo. On this system is a multi-beam sonar, and this enabled us to map the seabed to about 1 meter resolution. We are mapping an area that had previously been discovered, as well as some exploratory dives. In addition to the AUV, we were also using SuBastian, and instead of the basket you see coming in and out on the vehicle, we'd mounted a specialist mapping sled, which consisted of a LiDAR system, so a laser system for scanning the seabed, and also acoustic systems, and a photography system. This allowed us to map the seabed and take images to sub-centimeter resolution. Those maps then formed the basis of the second part of this expedition, where we went and investigated the known vents, as well as looking at the new vents that we'd found. Today we're visiting the vents that had previously been explored. If you joined us on yesterday's dive, you would have seen the phenomenal images that we got from the new vents. And you can check these out on Schmidt Ocean's website. You can also visit our blog on schmidtocean.org. Okay, the vehicle's now off the seabed. As we've just been sampling in the sediment, the co-pilot is rotating the wrist on there just to make sure there's no sediment that's got in there. And then we will be continuing with the science aspect of our dive. You'll just see in one of our cameras, the science camera, which is controlled by our scientists on board. So you'll see it moving around as they scan the seabed and look for things. In addition, we have several other cameras. Some are used by the pilots for flying the vehicle around, and others just give us information on gauges, etc. So these images coming to you from remotely operated vehicle SuBastian, operating at 3,686 meters, roughly 2 and a bit miles. The ROV has been operated off research vessel Falco, which is owned by Schmidt Ocean Institute. ROV operations make up about 12 hours of our daily operations. The other 12 hours is spent mapping the seabed. Falco itself is a great mapping platform and science platform. So in addition to SuBastian, we have a number of sensors that we can use. We've been mapping the seabed around here. After all, something like 10% of the world's oceans are mapped to a useful resolution, so we've been adding a little bit to that puzzle. Falco is equipped with a EM302 multi-beam sonar that allows us to map down to around 8,500 meters, so putting about 99% of the world's oceans within our reach. Here we've conducted about 150 hours of multi-beam mapping, covering about 1,400 nautical miles of survey lines.
Where would you like this one? Same spot.
Okay, in a moment we're going to give you a view of our control room. At the moment we're about to do another core sample, and in the small overlay that you see, momentarily you'll see the control room. Right at the far end down there we have Deborah, who's manning the data components of this expedition. To Deborah's left is Cody, who is operating the manipulators. You might just be able to see the small master unit that he is using on the ship to control the manipulators. Then to the left of Cody we have our pilot, Chris. At the moment Chris is just monitoring the vehicle. As we land on the seabed, we're slightly thrusting down to keep the vehicle in position while we actually collect samples. They're big, powerful arms, and if we push them into the seabed, we've got a habit of lifting up the vehicle. So Chris is just there making sure that the vehicle stays in one place. By here, to Chris's left, we have Victoria, who's chief biologist on this leg of the expedition. She's observing the current sample and pointing out interesting things that we should go and explore or collect. In the back of the room, which I'm not sure you can see, we have our team who are logging events. Basically, as we go through this dive, we want to log everything that we do, make a detailed record so that in the future, if further teams come back here, they can match our data and make sure and actually work with the data we've collected. Awesome. As you can see, there's a lot of screens. You're just getting one feed. We've got navigation screens. We've got... We'll go back to the sampling now.
Yeah, don't eat it. Bite down hard. Thank you. Oh, thank you. Am I still on? So you just got me smelling that rock.
I didn't know you were going to smell a rock.
At the moment, one of the rocks we collected is being shown around the control room, and one of the ROV pilots just gave it a good old smell, and obviously it's sulfur. It smells very nice. Nice rotten egg smell.
Full sensory experience.
So the samples you see that we're taking at the moment, they'll be brought on board the ship, and then they will be processed by our scientific team, and then they'll be taken back to their institutes for further study. Schmidt Ocean Institute is an open information organization, so once this cruise is over, our scientists have a period of time where they process the data, but in about 6 months this data will be made available to other scientists and other people who are exploring this area. At the moment you can see the core sample that we've just taken. The clear tube is now brown as it contains a large sample of sediment, and our co-pilot is placing this into one of the holders. Once they've placed it in the holder, they'll put a securing bungee over it. One thing that does happen occasionally is these may have gas in them. As we come up, if we don't and the gas expands, these cores can pop out. So one of the things is to restrain them, we put a bungee over there. And they also can bend through the top. Pilots can be quite dexterous with these arms. I mean, these things are incredibly powerful. I think they can give about 1,000 pounds of force onto something. So one of their trainings is to pick up an egg with the arm and make sure that they don't crush it. At the moment there's a discussion going on in the control room about what we're doing next, so we'll pan the camera around and have a look at some of the area. Here you see the sample basket with our various instruments. In front of you, lots of clams, bacterial mat, and on that note I will hand you over to a scientist who can explain the bacterial mat and the things that you are seeing. This is Leighton handing over to a scientist. Thank you.
Hello everyone, this is Dan Spett. I am a postdoc in the lab of Victoria Orphan, who is the chief scientist on this leg of the cruise. We are currently looking at a clam field where you see the siphons of the clams sticking out. These clams are typically associated with bacterial mat because they, rather than eating, feed on the sulfide that comes up through the sediment in the vent fluid. They feed on this sulfide using bacterial symbionts. So they have a special organ where bacteria that use sulfide and oxygen to generate energy and biomass grow and provide the food for the clam. Right now, center of the screen, you'll see an iridescent scale worm. Saw an iridescent scale worm. What we are currently trying to do is grab the temperature probe that is mounted on ROV SuBastian. The fluids that come up, hydrothermal vent fluids that come up through the seafloor in this particular area are about 280 degrees centigrade or Celsius, and where they come venting out of these towers. So by measuring the temperature, this gives us an impression or an indication of how much fluid, as compared to seawater, which is about 1 degree Celsius, or 1.2, how much fluid is at a certain sediment depth. And that gives us important information to interpret what we find in the sediment cores. Basically, it tells us how much food is available to the microbes that live in the sediment. You'll see we insert the probe first halfway into the sediment to get a reading at about 15 centimeters depth, see what the temperature is over there. That currently reads 27 degrees and is going up, probably stabilizes somewhere around 30. The ambient seafloor temperature is around 1.6 degrees centigrade. So you can see that even at a very shallow depth in the sediment, it's very comfortably warm there. And for those of you who have been watching for a while, you have seen that we melted one of these tubes that we just stuck into the sediment. These tubes are made of polycarbonate and have a melting temperature well in excess of 150 degrees centigrade. This was the first time that we cored a sediment, on this cruise that we cored a sediment that was hot enough to melt one of these tubes. As you look to the right of the temperature probe on the screen, you'll see that some of the sediment is whirling up there. That is partially through vent fluid coming up through the disturbed seafloor. It's not super clear on the screen right now, but as we pan to the right after this temperature probe has been taken, you'll see that some of the areas are the water is really shimmering. So you see the mixing of the vent fluid that is warm and has a different density than the seawater, and that way you can spot where the vent fluid comes out of the seafloor. The temperature probe that we're sticking into the sediment right now has roughly the same length as our core liners. So by first pushing it in halfway, we get kind of a midpoint temperature for the cores, and then pushing it all the way, we get an idea of what the temperature at the bottom is. These, where we're probing now, is the site of where this gray granular bacterial mat is. I don't quite know yet what the organisms are that live in the gray granular mat, or whether that texture is actually microbial or a mineral precipitate. But that's one of the things we'll find out. Regardless of what the surface looks like, the entire sediment will be filled with microorganisms that feed off the chemicals that are in the vent fluid. And in turn, these microbes provide the energy and the food for everything that you can see that lives here. So the temperature in this first core was about 29 degrees halfway and 46 degrees at the bottom. So not incredibly hot. Now if you see to the right, to the hole in the seafloor, to the right where the temperature probe is, you see some shimmering water. That is. The rain as we're zooming in on the mat, you see polychaete worms and really small red-colored copepods. These are probably grazing on the microbial mat at the top.
Yeah, slurping. Massive worms.
I just heard the term rioting mass of worms to describe what we see on the screen right now. These small invertebrates live white, not living in the guts there. They're their food. I get corrected. I'll now hand off the microphone to, as we're about to sample some of the invertebrates that live on this microbial mat, I'll now hand you off to Shana Grafferty, who is an expert at deep sea invertebrates, and particularly the ones that have microbial symbionts, and she'll explain more about the nature of the creatures that we're about to sample now.
Hey everyone out there, we are commencing suction sampling of this pretty unusual invertebrate community here. So if you had been watching, you were seeing us insert a temperature probe into this really hot sediment, measuring temperatures well above 90 C and even higher than that. And you see as we suction sample, quite a bit of oil is coming out of the seafloor. So this is a very strange community. And there are worms, so there are polychaete worms on the seafloor. There's a very unusual red snail we just spotted that looks new to our experts on board. And we're interested in what kinds of animal life can possibly live in these conditions. This is fairly extreme compared to what we would imagine would be a livable habitat. But of course they thrive in this habitat, and we're interested in how they do it. So we will be collecting some of these animals and measuring their capabilities based on their genome, and then DNA and RNA, just to see how they are possibly withstanding not only the hot conditions but also the toxic conditions. So these fluids are coming out very hot and laden with hydrogen sulfide, which should be poisonous to most animals, and yet here they are foraging on the microbial mat that has formed on the surface of the seafloor. So that's what we're up to here. You see a beautiful pear alvanellid just there in view, with its feathery red head, and we will suction sample them along with some of the other inhabitants.
Okay, and welcome back to Schmidt Ocean Institute's live feed. This is coming from ROV SuBastian, 3,686 meters below the surface of the Sea of Cortez. You're joining us as we are conducting sampling. At the moment we're using our suction sampler, which is basically an underwater vacuum cleaner or hoover. This allows us to suck samples up, and this device has several chambers, so we can store samples in each one. You can't see, but off camera there's like a revolver with 7 clear chambers. We suck the samples into one of these and then rotate it so we can keep the samples segregated so they can be sampled up on the surface. Reason for this? Well, some samples we don't want to contaminate from one jar to the other, and also some of our samples might try and eat the other samples. Yes, as I said, we're 3,686 meters below the surface. Now, some people have been asking, how come our temperature shows 2 degrees Celsius when we're this close to venting water? Well, it cools really, really quickly in the deep ocean. So it may come out at 280 degrees, or as we just saw recently, 200 degrees. But it cools within centimeters of the vents, well, a few feet. You may occasionally see the temperature on the vehicle jump up to a balmy 3 or 4 degrees Celsius. But in general, the heat dissipates pretty quickly. Although, those who were watching just now, when we put the probe into the seabed, it can be up to 200 degrees Celsius. So at the moment, you'll see your camera is moving back and forth. That's actually operated by our scientists on board the vessel, who are looking around, looking for specific samples that they want to take for documenting on board the vessel. Now, the good thing about a remotely operated vehicle is that in times gone past, oceanographic vessels in, say, the 50s or 60s did a lot of trawling to bring up samples. So we put a big net down and trawl the seabed and bring up samples. Obviously, they get damaged, and nowadays we know that trawling is rather destructive to the seabed. In the early 80s, ROVs started to become used for science, and they used, there's a number of vehicles around the world now capable of diving to some of the deepest parts of the planet. And the good thing about them, we can go down, we can look and document with our 4K and HD cameras in situ and get some really cool video and understand the ecosystems. But we can also sample. So unlike the sampling of the past, where we trawl an area, we can now go down with tools like the ones you're looking at the moment, the suction sampler, and suck up individual samples.
You want another parallel vanilla that you did with? It'd be great to get.
But obviously, one of the coolest parts of this is the video footage, and this will be analyzed many times after this. So what we see now, each time we look at the video again, we'll see more and more stuff, and this video will go to the archives once the expedition's over, and then other people can look at it. I will now hand you over to Okay, so much just brought to our attention that I've been reading off depths which are at the moment we're at 3,686. I've been made aware that the overlay that you guys are seeing is actually different. It is showing 37,000 or so? 3,700. Okay, so what we're actually looking at the moment, the depth is 3,686 meters. So apologies if the overlay showed something different. We're currently operating in the Sea of Cortez and Gulf of California. This is our last science dive. We've got approximately 1 hour of science left. This cruise is comprised of something like 22 dives. 3 of them were test dives, and the rest of the dives have been working around the known vents here at the site in Pescadero Southern Basin, as well as new vents that we discovered. At the moment, the team is sucking up some of the samples. These will be brought up and then sampled by the scientific team on board. Generally, our operations have fluctuated between conducting ROV operations during the day, and then in the evening we switch to multi-beam operations, which is a method of mapping the seabed 3-dimensionally. Falco is well equipped with this, and we have a system that enables us to map down to around 8,500 meters. So the 3,000 meters depth we're working in at the moment is an optimum depth for us to map in. Scientists are really happy with the sample that they've just taken. And off your camera, that's now gone into sample jar 3. At the moment, scientists are moving the camera and they're looking for more samples of interest. When they come out, they've got specific species that they want to collect and then study on board the ship to help them document them. Most people ask, have we found anything new on this expedition? Yep, we've definitely found a number of animals and things that our scientists believe are new species, but obviously a lot of that will be decided after we go back and they do genetic work on them and understand exactly where in the tree of life these creatures come from. So for those who are joining, the white is bacterial mat. This is bacteria that's growing on the seabed. And now from previous samples, we've seen roughly on the surface, it's several degrees. a little bit in is 25 degrees, and then about 20 centimeters in, we were getting readings of up to 200 degrees Celsius. So at the moment, our team is just stowing the suction sampler, the device that we were just sucking up samples with. Momentarily, I'm just being informed that we're actually going to take a rock sample now. So directly in front of the vehicle, the arm is going to sample a bit of the slab you see. Now the various tools are used for different reasons. The arm here is best suited for picking up the rock, but for some of the more fragile samples we use the suction sampler because we can just turn on a gentle flow, a gentle suck, and suck the samples up. The arms are quite dexterous, but for picking up soft samples they would have a tendency to damage the sample. Now bear in mind the co-pilot who is currently controlling the manipulators is actually controlling them from about 2.5 miles away. It's kind of a more complicated game than the ones you play at the fairground. These arms are capable of exerting over, I think, 600 pound, if not 1,000 pound of force. So our pilots have to be extra careful not to damage the samples. At the moment, our pilot and co-pilot are talking. You'll see that there's a tray coming out now. The camera's moving slowly back. The tray coming out contains a basket full of all different all different equipment that we're using on this dive. We have the cores that you've already seen. On the left is our titanium samplers, but at the back of the basket is 3 boxes in which we can put these samples as we pick them up. You'll see that the boxes in a minute, if the camera comes down, are labeled B1, B3 and B4. These are further compartmentalized into individual compartments, and as we put the samples in there, scientists on board make a note of where each sample goes. At the same time, you don't see all our cameras, they're also taking still shots and imagery of the samples so that when they come up to the surface, they know exactly which sample is which. In the back of the control room, we also have an event logging system, so images that are taken there are tagged and geo-referenced, along with also all the sensor data from the vehicle, so the salinity, conductivity and temperature. So we know the exact environmental variables in which these samples were taken. And the principal scientist has just turned around, Victoria, and said, "Can I give a shout out to Victoria's mum? Hello, Victoria's mum. You must be proud of your daughter." And it's a special day for us all on board Falco. This represents our 200th dive of remotely operated vehicle SuBastian. ROV SuBastian first came on for science use in, was it late 2016? So in roughly 2 years.
So just to say it, or is there a sample?
I don't know.
So at the moment, scientists are debating what we should actually do. We have 45 minutes of this dive left, and then we will be recovering the vehicle, and that will be the conclusion of this cruise. We'll be heading to Mexico, where we'll get ready for the next expedition. Obviously, this isn't the end for the science. This is just the tip of the iceberg. For 1 hour, I'd say, of probably science at sea, there's probably 20, 30, if not more hours of processing, writing up and documenting exactly what was done here. While we're waiting here and you're seeing that they're securing some of the samples, I'd like to say a big hello to Schmidt Ocean Institute shoreside team, as well as the team on board the vessel who are making the science possible with the vehicle and delivering the science you're seeing in this video. We also have a team shoreside who help plan and permit for these crews. So a big thank you to our team in Seattle and our team on the East Coast and West Coast. I'll also say a big thank you to our founders. Okay, and while we're waiting, we're just going to see if we can show you the location where we're working. You're actually seeing our vehicle control panel there at the moment. Oh, okay, sorry. We're about to hopefully send you an overview of where we're working at the moment. So, here we are in the world. There's the Gulf of California, and I think the Sea of Cortez. We're going to zoom in on the area where we're actually working at the moment. And coming into view now is the area where we've been working. This is multi-beam data acquired by both the vessel, AUV, and the ROV on the previous leg. We're working around some of the originally discovered vents. Also, the white data you see here is vessel collected data, so data collected by research vessel Falcor over the last few weeks of science operations. But we're focusing on this area here at the moment. And Deborah, if you could just show where the current vents that we're working around are in this location here. And if we zoom in, you can actually see the individual vents. This is really high-resolution sonar that has been collected by both the vehicle and the SuBastian and the AUV that we used on the first leg. We're currently in the vicinity of the big vent here. Currently, we are on the south side of it. And you can see it really stands out on this multi-beam. The vent itself, which this is a 3D construction from sound of the vent, is about 20, I think, meters high. And now we're going to switch back to our live feed. Oh, no, sorry. And now we'll be switching back to our live feed. I hope you enjoyed that. They're images that were constructed by, as I say, both our ROV using LiDAR and acoustic multi-beam systems, as well as the autonomous underwater vehicle that we use to map. So at the moment, our scientist, there's a small chimney there. I'm not sure if it's an active venting system, but we're going to head over and look and see what can be found on there. Victoria, our principal biologist would say, is currently explaining to the pilot about where she'd like to sample on this feature and the best way to place the vehicle so that they can scope out the species they're interested in. Some people ask why we study hydrothermal vents. Obviously, the main goal is to understand some of the complex systems that make up our oceans and exist underneath the ocean waves, and these are some of the most fascinating. But also, one of the reasons there's various organizations like NASA and JPL who are studying these features, because unlike things on land like the trees which use photosynthesis, these are chemosynthetic, so creatures that live around these can process the minerals and that, or the bacteria that's growing on them, and eat them, so they don't rely on sunlight. And now that's useful for when we consider the rest of our solar system. Some of the planets that are moons that NASA has been investigating have got reveal that there's water or water sources on the planet, and also some of them also reveal that there might be hydrothermal activity. So could some of those moons in our own solar system harbor life very similar to this? Who knows? But first of all, we need to study the life on our planet, and maybe one day this knowledge will be taken forward into our own solar system and reveal other life on other planets. But for now, we're studying one in the Gulf of California at 3,683 meters. You'll see the small tower coming in. Now this is a smaller hydrothermal vent. Some of them can grow up to 40-50 meters high, I believe. One of the biggest ones is called Godzilla, and they can be quite complex and amazing features. Large spires sticking out of the seabed, or chimneys venting really like hot jets of water. Some of the ones I think in the Cayman Rise area are venting over 400 degrees Celsius. So at the moment we're just looking at this vent and checking out some of the species that are on there. Our pilot's gently nudging the vehicle closer. A lot of these features are very fragile, so we try not to disturb them. Even a little bit of downdraft from the vehicle, just a little bit of push, can topple some of these. So it takes some delicate maneuvering of the vehicle to get it in place. I don't know, that looks kind of something like from Lord of the Rings. I was just saying that, it's like where Sauron's eye is. And you can see the real diversity of creatures that are on such a small vent. You've got your bacteria, there's like scale worms, tube worms, and other biology. I'd say hydrothermal vents were first really discovered and studied at the end of the 70s. And then almost just after that, in the early 80s, ROVs started getting used for more and more scientific and oceanographic work. Woods Hole were operating the small Jason Jr. from Alvin. And then since then, more and more remotely operated vehicles have made their way into science. We'll see ones like SuBastian. And then you've got Jason, the newest version of Hui's lineage. ISIS in the UK. And as you can see, the arm is attempting to sample some of this old chimney. Now, collecting old chimney also allows us to understand what the minerals were in the vents in times gone past. Gone by. But again, these arms are being operated by someone who's 2.5 miles away, sampling a rock of unknown consistency. For those who weren't on earlier, the arm is being controlled by our co-pilot on board RV Falco. The arm up here, we have basically a small replica of the arm which is on the vehicle. This is called the master, and the co-pilot is moving the various joints on that arm, and the arm subsea, known as the slave, is mimicking those movements. There's a button to close the jaws, and we can also slightly adjust the gains and the pressure that we put on these. But again, these are unknown consistencies. They could be hard, they could be crumbly. We really don't know until we sample them. As with all sampling, we do this in a conscious way, making sure that we don't damage unnecessarily. And you can see the arm is just gently exerting pressure. And that looks like a pretty large sample that we've just got here. Now, as with anything, we don't count eggs before they hatch, and hopefully we'll now put out the basket and try and collect this sample. Looking at some truly amazing imagery here from 3,684 meters below the ocean waves here in the Sea of Cortez. remotely operated vehicle SuBastian, operated by Schmidt Ocean Institute, off research vessel Falco, is currently sampling a small chimlet. Now, a team of ROV pilots, and all come from various backgrounds. A lot of them have experience in the science sector. Yeah. Principal scientist has just pointed out that our pilot was taking a picture of the sample, and you said when the pilot's taking a picture of the sample, you know it's a good sample. So now we're just gently going to try and get this in the basket. To say with everything, there's a lot of unknown quantities when picking up samples in the depth. As you can imagine, as we're doing this, there's about 15 pairs of eyes staring at the pilot and co-pilot, just willing the samples to get in the basket. Okay, you just saw the perspex lid of one of our boxes open. These are hydraulically actuated, and now we're going to try and place this large sample we've just collected in the central basket. Our cameras are usually operated by the scientists, but now have been switched over to the pilot and co-pilot who are using them to make sure we get this sample safely in the basket. Oh yeah. So in order to conduct science here, we have to apply for permits. We can't just turn up and actually sample anywhere in the world. So what we're doing here, we have international collaboration between US, I think Australian as well, and Mexican scientists, and they're all working together on this. So when they initially do the proposal as well and submit it, they request permits from the Mexican authorities, who obviously we're working with our Mexican counterparts, and they review the proposal from the scientists. They're happy with it. They issue the permits that allow us to go and collect scientific samples and scientific data. We can't just turn up and start taking these. And these permits are strictly controlled on what species we can take and what level of work we can do. As well as our permits for this, there is international standards for operating around hydrothermal vents. Just examples, some of these like the Interridge Code of Conduct. And we use these when working around this, so we don't unnecessarily sample. We make sure that everything that we sample is shared with the scientific community. And I'll be quiet for a second while we make sure we get this sample in the basket. It's quite tense here in the control room at the moment. There's a lot of people focusing on the pilot and co-pilot roundabout now. And there we go. That was a phenomenal sample. And that was picked up by our co-pilot, Chris, who's operating the arm from 2 point. Yeah! Everyone was holding their breath until the lid of that was closed. That was an absolute phenomenal sample. We obviously were aware of what we're sampling, but that was more cheering the co-pilot for a fantastic sample. As I was saying, back in the day, in 20, 30 years ago, or maybe 30, 40 years ago, a lot of sampling would have been done through trawling. The sample you just saw was a tactical sample. We can now go down with a vehicle, pick up an individual chimney and put it in the basket, bring it up almost totally intact and study it. Obviously, in the days of trawling, it was quite destructive to the seabed. We now know what sort of damage that causes. So the ability to use a remotely operated vehicle, go down and directly sample something and observe the environment is a fantastic tool for our scientists. We're now entering the last half an hour of this dive. If you have any questions, you can ask them on our YouTube feed or on our Facebook feed. And I will now hand you over to our principal scientist, Dr. Zurenberg, or Professor Zurenberg, okay?
Hello everyone, this is our last dive. I have not been listening to what Leighton has been telling you, but I think he's got you pretty caught up on what we've been doing. I'll tell you that from my point of view, this has been a really successful cruise. We've achieved all of our goals. We are now going to try and get some of these little purple scale worms. We're going to slurp some of those up. And then right after that, we're going to be moving up to the sort of the base of the chimney. We're going to collect some big slabs of rock. And then as we fly out, you'll see some of the high temperature vents up at the top. So we've got only about a half an hour left on the seafloor here, but you'll still see some more stuff. So I'm a geochemist. I'm from the University of California at Davis. I was on this at this site with the Mbari ship, the Western Flyer, using their submersible, the Dock Ricketts, in 2015. Mbari led a cruise. Mbari is the Monterey Bay Aquarium Research Institute. They led a cruise to an ocean spreading center just a little bit south here called the Alacarn Rise. We did some very detailed mapping and sampling of that, and then our last dive on that cruise, we had sent the AUVs, the autonomous vehicles, up into this area, the Pescadero Basin, and they had done some mapping, and we got an unprocessed map just days before we were scheduled to dive, and the map showed these large chimney structures that you're seeing, and they showed a temperature anomaly. So we had a very good idea that we had a new vent field, and so our last dive, we came up to this site to try and find that. We had water sampling bottles aboard, so we were very excited to try and get vent fluids and see a new vent field. And that had been about a 2-week cruise. Every day was just perfect weather until the morning we got to this site, and the wind was blowing and we couldn't launch the submersible. We also had to be into port that afternoon or the next evening, so we had a very strict time limit as how long we could be here. So I remember very distinctly knowing that we were sitting over a potentially new vent field, ready to dive on it, and the weather just not being cooperative. So we sat there. The captain said, well, we can't wait any longer than 10 o'clock and we have to leave for port. 10 o'clock comes, the wind's dropping a little bit. Captain says, well, maybe we can wait till 11:30. 11:30 comes, the wind's dropped a little more. I think we finally launched about 12:30. We had 3 hours of bottom time to explore this field and try and find some place where we could fill up water bottles. And so very sort of exciting but intense 3-hour dive, seeing this vent field for the new site, getting some water samples in one vent, and then having to pull off the bottom and head for home. So having seen this vent site, I've been working on these kind of deposits for a very long and productive and happy career, as have the people we're with. And I think all of us who saw this vent site recognize the importance of it as a potential site where a lot of questions about hydrothermal vents could be better answered here. One of the reasons is this is a very large site, and we think these are very old deposits. There's a lot of material deposited above the seafloor. We can see that these mounds are growing up and falling apart. You'll see as soon as we come up on the side of this chimney that there are collapsed chimneys and pieces of what we call flanges. You'll probably see a flange when we fly out of here that's active. And so these things are falling apart, and the calcium carbonate of which they're made is also unstable at these depths. They're dissolving. So the fact that we have these big deposits mean that even though they're forming rather slowly, they're still growing faster than dissolving. And given the amount of calcium carbonate deposited in these big mounds, we knew this had to be a very old site. And also because we've done, when the AUV flies and makes the map, it does seismic reflection in the upper part of the sediments. And we can see that the sediments are actually on-lapping this. Given that the sedimentation rate is very slow, that means that, again, this deposit must be very old. The reason we want a big old deposit, if we want to understand the sort of the chemical and heat flux, we need to drill to the subsurface to really get to understand what is going on to drive this. That's a hard process, and hitting a specific target when you're drilling is even more difficult. So if you have a large deposit, that means that underneath it is a large area that's been hydrothermally altered. So that makes it a much more easily obtainable target to hit. So we feel like this is big enough that we could bring a scientific drill ship in and investigate the subsurface. Also, because we have fairly soft sediment and relatively soft carbonate, it should drill much easier. So we think this is a site that could become a future reference site. With the new maps that we've made, we're hoping that the rest of the community will recognize the potential for this site, and it will attract other researchers here. And our long-term goal, I think, is to try and to make this a site of future study, because I think there are really a lot of questions that could be answered here that can't be answered anywhere else. So our goal is to look to the future, and this cruise has really put us in a much better position to attract other researchers here, open it up to the international community to think about this, to give us, once we process some of our data, to allow us to write proposals to bring new ships here. Our colleagues from Mexico have planned to bring their ship, the Alpha Helix, in here to map, and so this is sort of the long-term goal.
All right, so we just collected a nice sampling of these scale worms, and there's the little blue and pink organisms around that hydrothermal vent chimney. These are indigenous to this particular area in the Pescadero Basin, and so we're interested in getting enough of a sample that we are able to understand a little bit about their morphology and genetic makeup. So that will be a nice sample set for our co-op PI on the cruise, Greg Rouse, to do some additional work with those organisms. We're now heading up to the Z Mound, where we're going to collect some additional pieces of the carbonate chimney. We're sort of reaching near the end, last 20 minutes of our dive, so we're trying to beat the clock here and get some last remaining samples of the expedition. So my co-chief Rob is trying to direct the pilots right now to the best spot to do some quick sampling. If you have any questions, please send us your notes by YouTube or Facebook, and we'll try to answer them. And thanks for watching.
Okay, so we're in the last 15 minutes of this dive, and our principal scientist is going to take us to another position for some final sampling. You're joining us from research vessel Falco, operating remotely operated vehicle SuBastian, 3,670 meters below the surface of the Sea of Cortez. We have conducted something like 20 odd dives on this cruise, so I will just give you time. Yep, we've done the best part of 203 hours of ROV operations. Now, because of the depth, that equates to about 130 hours on the seabed. The rest of the time is transit to and from the seabed. Takes us roughly 2 hours. Takes us roughly 2 hours to get up and down. I'm now going to hand you over to our principal scientist.
You're live.
Okay, we are now coming, we've come up the Z mound, and as you saw, there were big blocks at the base when we came up, and now you see this big fracture through the mound right in front of us. So these mounds are growing up and falling down, and you see some nice flat slabs in front of us. Those are what we call pieces of flange, and we're going to try and sample some of those. And rather than explain exactly what a flange is, after we collect these, we're going to fly underneath an active flange, which is really going to be spectacular. Off to the left part of the screen, you see that hollow round thing. That's a piece of chimney that has fallen over and now has been partly incorporated into the mound. So these things are always growing up, falling down, growing up, falling down. They're really, quite active features and somewhat unstable, but we don't know the time scale very well on which this happens. So although we see these things, growing and crumbling, they're somewhat ephemeral. We think this is old, but it's hard to put a quantitative number on just what old is, because we have very few ways of really dating these. There just aren't very many good radiometric clocks that work on these kind of rocks. We think that the best way to get a handle on the long-term age of these is to actually come and core the sediment that's around them, and that will record the hydrothermal history and the sediment layering. That's something that we hope to do in the future, but we haven't really done yet. So that's one of our future plans. So that's a piece of flange, and is in contact with 290-degree fluid when it formed, and the top is now upside down. And so what we hopefully will see when we fly out of here is that sometimes, the top of this big mound, there are chimneys like you're used to seeing venting hot fluid, but sometimes the fluid will find a crack in the mound and start coming out, and you basically start growing a shelf that grows outward from the chimney base. They grow by the mixing of hydrothermal fluid, which is high in calcium but has no sulfate, and seawater is rich in sulfate, and when hot calcium-rich fluid meets cold seawater sulfate-bearing fluid, then it exceeds the solubility of calcium sulfate and forms a mineral called anhydrite. And the anhydrite starts to grow out in that mixing zone, and so where the fluid comes out from the crack, you start to form a little shelf that grows out, and as it grows out, hot fluid ponds underneath it, and the hot fluid itself can't form anhydrite because there's no sulfate in it, but you start to cool that fluid and you form calcite. So the white part of this sample that just went in the box is calcite, and the bottom edge of it had 290-degree fluid ponded underneath it, and the top part had some anhydrite. So we're going to probably collect a couple more samples here, and then as we fly out, we're going to try and move around the other side of the chimney and see an active flange. So if you look just to the top of your view, out in the far distance, you see that white flat shelf. That's a flange that has hot fluid under it. We're going to look at that on the way out, and you'll see how these things form. Because the hot fluid is ponded, it's buoyant, it wants to come up, but it's ponded under these shelves. it's in contact below it with cold seawater, and the interface between them is incredibly sharp. You'll have a gradient from 290 degree fluid to 2 degree centigrade seawater that occurs over millimeters, maybe even less than a millimeter. So it's a very sharp interface. And the refractive index of water, or the speed of light in water, is a function of the temperature. So when you see shimmering water, it's shimmering because the hot water has a different speed of light, and so it changes the way light comes. Well, when we get underneath this flange, you'll have this really sharp interface between cold water below and hot water above. That's such a sharp interface it actually can reflect light, and it acts like a mirror. And you'll see this hot fluid rippling along, and if we come in the right orientation, you might actually see the reflection of the submarine reflected off of this hot pool below. So that's what we're hoping to do on the way out. So right now we're just picking up pieces that have fallen off of previous flanges. We'll take these back and use them for science, and then we'll try and give you a good view of the active flanges on the way out. Okay, so now you're looking at one of these flanges again. From having studied these, I can tell that this is a piece of flange that, again, has landed upside down. So the flat part on the top that's very smooth is where the hot water was ponded. The white material is calcite that was growing down into that pool, and the grayish, rougher material on the top was the anhydrite that formed the flange. And so this flange fell off of the mound and is sitting there now, and you can see similar things in the distance. And we're going to try and get one more sample here, and then we're going to have to leave bottom, but on the way out we're going to try and fly right up under an active flange to get a good view of what these look like. You can see that these things are pretty solid. The calcium carbonate is a pretty tough mineral, so we're kind of manhandling this, trying to break off a piece that will fit in the submersible. We can't really take the whole thing home because there's not room in the boxes for the whole thing, so we're trying to get manageable size pieces. So being an old geezer, I remember something from the 60s. There was a book that came out, and I think the title was Never Eat Anything Bigger Than Your Head. We may have just violated that. So I think you can see that when the pilots really want to do something, they can get rather creative. And we're going to find a little slot for this treasure and bring it on home with us, we hope. All right, so now we're lateraling around. We're a little more toward the base. Now you see lots of big blocks of talus have fallen off of this mound. And this is how these mounds grow. Chimneys grow up, fall down. Hydrothermal fluid comes up and cements it all together, and over thousands, tens of thousands, I wish I knew exactly years. This is how we build up these deposits. In the distance, you can see where it's a little white, and that's where the most active fluid flow is. So on the top of this structure toward the top, there are lots and lots of tube worms and active vents, and we're going to move over and try and do a vertical transect up this structure and get a good look at the hot fluids. There's an isopod that's just flying by with the long legs. He just ran into the camera. He thought that was a little rude, so now he's leaving. So now you see some worm tubes down here, and then in the distance you see the whiter areas and the bluer areas. Those are areas where there's bacterial mat, and you're going to see live tube worms. That's where the fluid is coming out. Zoomed in now, you see little bits of there's a Riftia tube worm right below us. There's a red plume. Most of the tube worms here are a species called Oasisia. If you're used to looking at vents, you've seen a lot of Riftia. There are very few here, and we just got a quick peek at one of them. Out in the front is weakly active area. The blue and the white is mat, bacterial mat, where there's some leaking flow. You see big pieces of flange below us. And now we're maneuvering around to come up and see the active top of this chimney. Okay, now you are looking at an active flange, and so in the distance you can see there's a flat black surface. That black surface is actually the reflection off of the hot water that's ponded under there. As we come into this, we'll get a nice view of that. So you get an idea that of looking at this. There's a nice ctenophore coming by to see us on our way to the surface, and now you're seeing in the distance a beautiful clump of tube worms. Back in 2015, this was one of the first things after driving to this mound, when we got to this mound, this was one of the first sites we saw, and we were all really pretty excited by it, and I still am. You just see how much shimmering water there is. So now we're up to the active part of the mound. And so these flanges just grow out, and then at some point they just become unstable. They fall down, and that's what we were sampling at the bottom. So. Okay, and now that we're up above this hot water, we want to take a sample of the fluid, and we have something called Niskin Bottles. The little blue stopper you see coming out of a plastic bottle is a Niskin, the one that we've already fired. And what the pilot's going to do is, when we get over the shimmering water, he's going to pull a little tab, and that's going to allow the next bottle over to close and just trap some of the seawater, and then we'll be able to analyze that and see the water composition and look for microbial indicators, filter it, look for larvae of the vent fauna. So one of the last things we're going to do is fire that Niskin, and then we're going to have to leave this site for this year. But we all hope that we and others will be back at this site. A beautiful place, a great place to do a lot of science, and we're hoping that this is the beginning of more studies here, not the end of studies. Okay, so the arm is moving in toward the submarine. You see those little rings. We're going to pull on one of those. So he's grabbing the handles that are connected to the rings. You see there's a wire connected to that ring. That wire goes up to the Niskin bottle. And now he's grabbing that, and he pulls that. The Niskin should have just fired. That means it will close up and trap some of the seawater that we want in there. And you can see there's a little tab that says number 1 there and is now closed. And between 1 and 2 you see an open one. So we've just pulled the tab so that flap goes closed, and now we've got that sample. And now we're going to fire another one just to have more sample which to deal with. And now we pan back up, and number 4, which is just out of the view, has fired. So now we've got that fired. So we're now about to leave the seafloor.
Down here at the bottom, so we'll start to the top. Okay, we'll let you reps get on the drum and then start greasing.
Okay. So we bid you. We're ending the dive. This is your last view and our last view of the seafloor. We bid you a very fond farewell from the RV Falcor, and I would like to express my sincere thanks to the ROV pilots, the crew, the ship, all the people who made this possible, and especially the Smith Ocean Institute for this opportunity to do science.
All right, just want to sign off and also send out my sincere thanks to everyone. This is Victoria Orphan. We've had a fantastic team of scientists on board, an interdisciplinary group of people ranging from geophysicists to biologists to microbiologists and geochemists and engineers. And this collectively has made for a really interesting and fantastic opportunity here for the past 2 weeks in Pescadero Basin. So it is with a little sadness we're looking at the top of the Z-mound sort of disappearing in the distance here. It's really one of the most remarkable sights that I've ever seen in all of my time doing research in the deep sea. So it's we feel very privileged to have had the opportunity to work here, and we're really excited to get back to our respective laboratories and analyze all of these great samples. So thanks for following us. This is Victoria Orphan and my co-chief Rob Zurenberg and Dave Caress. And we're going to pass it on to Dave to also say a few words at the end here.
Okay, this is Dave Caress, co-chief scientist of the first leg, the mapping leg, and on behalf of the Monterey Bay Aquarium Research Institute, or the MBARI team that came out on the first leg and conducted all the mapping work. I'd also like to thank the Schmidt Ocean Institute and all the folks on shore with SOI, and particularly out here on the ship, that have made this possible. For us, this expedition has been very successful. We've between the mapping AUV, meter scale surveys, and the low altitude centimeter scale surveys, we really succeeded in achieving our goals in terms of extending the coverage of what's known around the South Pescadero Basin and identifying previously unknown vent sites, and enabling us to come in then with the ROV and investigate those. And with the low altitude surveying, we covered much of the Auka vent field, the previously known vent field, and worked hard to make those data usefully available to help guide the biological studies here on the second leg. And we've, I think, succeeded in great part in our aims, and learned tremendously about where to go with all of this technology development in the future. I'd particularly like to thank the shipboard folks here on the Falcor. This is a tremendous ship. Many of us have sailed on a lot of different oceanographic research vessels over the years. And this is, I think, in many ways, a uniquely capable group of people that operate this vessel and this ROV. And I thoroughly enjoyed the last month being here on this ship and working with these people. So again, on behalf of the Mbari team and the whole science party for the entire expedition, thank you to everyone at SOI, and to all of you that have paid some attention to what we're doing out here. Thank you, and we'll bid you adieu as we now recover the ROV from its 200th dive, and try and gather all of our data and our samples and get prepared to